next speaker is Dr. Laskay, and he's going to talk about cell physiological effects of reduced deuterium content in the leaves of Elodea canadensis. I hope I said it right.
Good afternoon, ladies and gentlemen. I have come from Szeged, and perhaps for those who are not familiar with the geography of Hungary, it's worth having a look at the map. Here we are now in Budapest, and if you travel about 170 kilometers to the south or southeast, you can come to Szeged. But if you travel a little bit more, about 200 kilometers, you will get to the border. So Hungary is a relatively small country, and 200 kilometers can be considered as large distances. It's a relatively nice university town. Here in this place we have an open-air theater all summer, and this is the building where the high-rank officials and the rector of our university have their offices. And the population is about 160,000-70,000 people, but we have 30,000 students, and so you can appreciate how lively the city or the town can be, especially in term time. We often say that the whole town is a university campus. So anyway, we all know that plants are beautiful creatures because they produce food for us, either directly or indirectly, and also they produce oxygen for us, without which we couldn't liberate the energy which is stored in the food molecules which we consume. But of course, but in addition, plants provide us with interesting experimental system with which we can study basic cell physiology, and for example we can study the effect of deuterium depletion. Especially good or promising experimental objects are the aquatic plants, which are in close connection with the surrounding water, and they do both photosynthesis and respiration with the constant contact of their ambient water. So if they photosynthesize, they take up CO2 from the water. If they respire, they release CO2 into the water. So they might be useful objects and experimental systems for the studies of deuterium depletion. Perhaps not all of you are familiar with what Elodea is, but thanks to the morning session, we know that Elodea is a favorite aquatic plant, and is a submerged microphyte which now is present also in Europe. It originates from North America, and there were times when it was not a very nice guest arriving from the United States. In the 50s and 60s, it caused considerable problems in the waterways, many waterways of Europe, because when it brought into Europe, into Germany, it coiled around the screws of the boats on the River Rhine at the good old days when there was some wildlife in the River Rhine, and it caused stoppages and tie-ups. And in Hungarian, its name is átok-ínár, which means cursed water weed, and perhaps some of these problems can be associated with this earlier behavior. But now we can find this plant in pet shops, and all those people who have aquaria at home are familiar with it. Apart from this, this plant is very easy to work with, because the leaves can be stripped off very easily and can be put into any kind of experimental media, and then the physiological actions can be easily studied. this is actually this plant, which is a useful object for cell physiological studies as well. Have lots of chloroplasts in the cells, and all kinds of cellular studies can be carried out with it. So, as you know well, the plants can carry out photosynthesis and respiration. In the light, they carry out at the same time, both of them. But photosynthesis usually predominates, and in the light, plants are capable of fixing CO2 and producing sugar, and they are capable of utilizing the energy of photons. In the dark, they only respire like us, and with respiration they produce energy, ATP, so the plants are the only creatures which can carry out both different kinds of energy supplying cellular mechanisms. So the chemists were able to measure how CO2 can dissolve in water, and they were able also to measure the rates of the different reactions which are taken place when CO2 gets into water. And according to these measurements, there are different components which are formed when CO2 is dissolved in water, including carbonic acid, which can dissociate into bicarbonate, which can also dissociate into carbonate. But according to the speed of these reactions, it can be considered that at or near neutral pH values, this reaction is sort of fast. This reaction is very slow. This predominates only at high pHs. And so, therefore, the rate-limiting step is this value, this reaction. Therefore, for the sake of simplicity, at around near pH values, we can consider the dissolution of carbon dioxide in water according to this equation. And the three important components, the presence and the ratio of the components is a function of the pH, and you can see the fraction of CO2, carbonate and bicarbonate and carbonate in the function of pH, and this is the pH around 6.5, which we chose for our studies because that's the pH when half of the carbonic acid or half of the carbon in the water is present in CO2, and half of it is present as bicarbonate, so that the plants can take up carbonate without any problem. And if we put the plants in the water and switch on the light, they will start to photosynthesize and they will take out the carbon dioxide, and this will change the equilibrium, of course, and according to the basic rules of equilibrium kinetics, the reaction rate of the backward process will increase, and this will cause a decrease in the proton concentration. In other words, in the light, if the plants start to photosynthesize, we can expect a decrease in the pH, decrease in proton concentration, so an increase in the pH, and the reverse reaction is expected in the dark. So, what we saw when we did the experiments with these plants, these experiments were only in normal water, and this we will compare with deuterium-depleted conditions. So if we put plants into normal water, they will start and switch on the light, they will start to photosynthesize and they will increase the pH, and this pH increase is associated with a concomitant decrease in the carbon dioxide concentration, and the reverse process applies to when we put the plants in the dark. So when we did the same experiment in low deuterium, 87 ppm deuterium concentration, we, instead of seeing this gradual increase in the extracellular pH, we saw a gradual sudden drop followed by a gradual recovery. And when we did the associated measurements on carbon dioxide concentration, we saw a slight decrease in the carbon dioxide uptake from the water, because this is the uptake due to the photosynthesis. And in deuterium-depleted water, this uptake in the first sort of half an hour, it was decreased, but after that the rate of the uptake was similar to that of the control. And when we did the experiment in the dark, so that the plants could only respire, then the normal plants exhibited a decrease in the pH, which was a little bit accelerated in low deuterium water, but the acceleration also happened in the first half an hour. And the associated carbon dioxide measurements showed that this decrease in the pH correlated well with an increase in carbon dioxide content of the water, presumably because of the stimulated respiration. So from these studies, we have seen that in the first couple of minutes, or in the first half an hour, when the plants encounter low deuterium conditions, they show attenuation in the photosynthesis rate and a stimulation of the respiration rate. However, these two actions are not enough to account for the huge acidification, which, as you remember, we could see in the first half an hour when we measured only the pH in the presence of light. So there could be or might be something else going on, which might be an additional factor in causing this significant external acidification. And to find it out, we suggested that might be the membrane potential could be an important factor. As we know well, all living cells develop voltage across the plasma membrane, and this membrane potential is due to different factors. For example, the unequal distribution of the ions across the plasma membrane and the unequal permeability of the ions across the plasma membrane, and the presence of negatively charged proteins in the cytoplasm, and the presence of electrogenic pumps in the membrane, which contribute to this voltage. For example, the animal cells can be schematically simplified with this model that they contain relatively high concentration of negatively charged proteins in the cytoplasm, and this is roughly compensated in molar terms by sodium ions in the external medium. And if the plasma membrane would be impermeable to sodium ions too, as it is impermeable to the proteins, there would be no problem. But unfortunately, this plasma membrane lets some sodium ions into the cells, into the cell interior, and therefore the animal cells have have a sodium-potassium pump in their membrane which compensates for this effect. Otherwise, the cells could not maintain their normal volume. So the animal cells have a sodium-potassium ATPase which pumps 3 sodium out and 2 potassium in at the same cycle of the functioning of the pump. Therefore, it contributes to the existing membrane potential. Plant cells do not have this pump in their plasma membrane. They have this proton ATPase, and they also have cell wall. And it is this proton ATPase which contributes to the membrane potential in plants. And this membrane potential traditionally can be measured with electrophysiological techniques. So if we take 2 electrodes and put or introduce one of them into a living cell, we can measure current, and the source of the current is in the membrane, because the membrane is the part which separates the charges, and the membrane is the part which separates the 2 aqueous solutions. So the membrane is the source of the voltage, which is the important part in maintaining this current. And the interior of the cell is the cathode, and that has important implications, because nowadays, fortunately, we can use alternative methods to measure membrane potential. We don't need to impale electrodes into the cells. It would be extremely hard to do it with plant cells, because they have a relatively rigid cell walls. But we possess a range of fluorescent probes which can be of help, because they can penetrate into the living cell according to the actual membrane potential, according to the actual voltage. And one of these dyes, which is a fluorescent dyes, a potential sensitive fluorescent dyes, can be seen in this picture. And this is actually a lipophil cation. And since it is a cation, it is relatively easily taken up by the cells relative to the membrane potential. So the higher the membrane potential, the higher amount of this dye is taken up during a given period of time. So that's the principle of the measurement. If the membrane potential is low, there is only a little amount of dye which is taken up. If it is higher, we can get more dye in the cells, and if the potential is even higher, we will have even higher amount of dye in the cells. And since it is a fluorescent dye, we can measure the fluorescence which comes from the cells, and therefore we can estimate how big the membrane potential is. So we did this experiment with low D water, and what we saw was that in the first half of the experiment, in the light, the plant cells took up a huge quantity of this dye, and after that the rate of the dye taken up was relatively similar to that of the control. And that can explain, at least partly, why we observed this relatively significant external acidification. So, in conclusion, we can say that the plant cells are capable of sensing the sudden change in the deuterium concentration of the ambient solution, and the basic cell physiological processes show a slight change, and these changes focus on both of photosynthesis and respiration. At least we concluded from the attenuation of the CO2 uptake and the stimulation of CO2 production. And we also saw a triggering of hyperpolarization, presumably due to the activation of the proton ATPase. Important is, or one of the most important points, is that all the observed reactions are of transient nature. They are present in the cells in the first half an hour or first couple of minutes, and after that the cells gradually get accustomed to the new conditions. So the conclusions which we can draw is that aquatic plants are suitable experimental objects to study deuterium depletion, because they show a rapid and relatively significant change in their physiological pattern. And we think that this change in the cell physiology might be attributed to the change in the hydrogen over deuterium ratio, but we also saw that the plants quickly adapt to the new conditions, but these early actions can be exploited to study further what deuterium depletion might be doing in cells. Thank you for your attention.
The talk is open for questions. Go ahead. Can you wait for the microphone? Just 1 second.
Have you tried to estimate for the overall effects of the immersion in DDV of the plants? How long did you keep them?
No, that's the point, and that's why I asked you in the break that how long time did you need when you observed the increase in the growth rate, because we concentrated only this first hours, and frankly speaking, we didn't see any longer actions, and you gave me a very good indication that we might do that.
And are there any possibilities of chemically blocking the hydrogen transport through the cell membrane, the vegetal cell membrane? Because I don't know.
There are compounds which can be used in isolated plasma membrane vesicles. For example, vanadate, sodium orthovanadate is one of them. But I'm not aware of any studies which show that they work in vivo as well. But maybe it's a textbook story. We have to find it out.
I'd like to ask you a question. Do you think the fast adaptation, because this is how I would call it, to deuterium depletion in the plant system is consistent with the fact that plant enzymes, for some reason, especially the ones that are involved in carbon fixation and eventually photosynthesis, are just more discriminative of isotopes, meaning because we do know that although there is no isotope discriminating effect in mammalian systems as far as intermediary metabolism go, if we apply the same concentrations of stable glucose, stable isotope labeled glucose, in plant systems there is, in fact, discrimination against the heavy glucose isotope. Do you have any idea or any answer why this may happen and why there's different enzyme responses in plant versus mammalian systems?
Well, I don't know if we can say anything at the enzymatic level. But what we think is there might be a balance between photosynthesis and respiration in plant cells, because environmental factors usually affect them antiparallel way. And it's tempting to speculate that this this switch, which can find the balance between the two processes, might be the one which is sensitive to hydrogen and deuterium. But the exact nature of this mechanism is not known.
Last night, as I was just finishing my conclusions, I was really almost tempted to go into this metabolic contra-analysis field, which I'm not the expert in. We do have a collaborator from the University of Barcelona who is really the main guru in the field. And I was reading the book chapter we wrote together. Obviously, she wrote the part which relates to this enzyme elasticity, meaning that there are an elastic component of the enzyme function, meaning that in certain environmental factors, and probably deuterium depletion is one of the factors that can affect the kinetics of enzyme reactions just based on whatever else is needed, for example, a hydrogen for reducing, yet it's not really the main function of the enzyme to separate hydrogen in water, but actually uses that hydrogen molecule to, for example, synthesize fatty acids de novo. And even though there's a main regulation from the substrate point of view, as far as what the substrate availability and what the product level, positive, negative feedbacks on enzyme allosteric regulation, it seems that actually deuterium or hydrogen, which is practically just the other reactant in the reaction, but not the carbon source, can dynamically change the kinetic patterns. And this was just one area that I have been I was not ignoring, but I'm really not truly the expert, but we have collaborators who allow us just to go and get or gain more insight into these fields. And I'm just talking because we have a few minutes, so and I think it's really interesting. But based on your studies, it seems to me that actually plant enzymes have even more of an effect of this sort.
Yeah, that reminds me that in the first talk, Gábor mentioned that there are differences in plants carrying out different pathways of photosynthesis. For example, the C4, C3, and CAM pathway respond differently. Yeah, is this correct? Yeah.
Do you have any comments on that?
Regarding that, so the C, if it depending on what type of food we eat, for example, pea or popcorn, it could be more depleted or less depleted for deuterium, the carbohydrate, and the CAM plants even can enrich the deuterium, so depending on which metabolic pathway is present in the plant. So it can discriminate. The algae can discriminate in light between hydrogen and deuterium, but not in dark. So this is what I say, that it is present, I think, in the living organism.
Actually, this is how we know where the marijuana is coming into Los Angeles, because if it's from Colombia, it has a different deuterium and carbon enrichment than if it's from Mexico. Yeah. I mean, it was grown in Mexico. Yeah. We know because we work with reference laboratories, just for that particular purpose. So it's very clear. Yeah, it's very evident.
Yeah. Yeah. Okay.
Thank you.
Thank you very much. So.